Are you guys also with the YouTube open? Just
I am, Gustavo.
cool. So you give me a shout as well, just to let me know that's working in your computer as well. I think it's getting on.
We are already live, Gustavo.
Cool. Okay. All right. So welcome everyone to the next section of our graduate course on cell death and immune system. This is a great pleasure to introduce you the next speaker, Lorenzo Galluzzi, who actually has a bunch of appointments in very different universities around the world, in Paris and in Italy, in a couple of cities in Italy. But he's broadcasting from his major appointment in New York, where he's currently assistant professor of cell biology and radiation oncology with the Department of Radiation Oncology at the Weill Cornell Medical College, and an honorary assistant professor adjunct with the Department of Dermatology at the Yale School of Medicine in New Haven. Lorenzo is best known for major experimental and conceptual contributions to the field of cell death, autophagy, tumor metabolism, tumor immunology. In particular, he provided profound insights into the links between adaptive stress response in cancer and the activation of a clinically relevant tumor targeting immune response in the context of chemotherapy and radiation therapy. Lorenzo has published quite a bit of papers, around 500 scientific articles in international peer-reviewed journal, and his current age index is only 118, which is a shame for me, I have to say. According to a survey published in the Lab Times, Lorenzo was the 6th and the youngest of the 30 most cited European cell biologists in the period of 2007-2013. And he has been nominated highly cited research by Clarivate Analytics in 2016, 18, 19, and 20. Lorenzo currently serves also as an editorial position in multiple journals, including editor-in-chief of Oncoimmunology, International Review of Cell and Molecular Biology, Methods in Cell Biology, and Molecular and Cellular Oncology, and also associate editor for the Cell Death and Disease, my favorite journal, I have to say. And aging and the eye science. So he is far the best person to teach us immunogenic and non-immunogenic or tolerogenic modes of cell death. Lorenzo, thank you very much for sparing time and to be with us today.
Thank you very much, Gustavo and Karina, Ricardo, and the organizers of this class and this course. I understand it's been going on for a few days now. Unfortunately, I was not able to connect with you previously. And thanks for the extended introduction. So what I want to do today is give you a large overview of what I've been doing from conceptual and experimental standpoints over the past now 15 years. It's been a long run, but the first time I heard the word cell death was when I moved to Paris in 2005. So you can tell it's been quite a while as I digged into this topic. And I wanted to also apologize up front because I'm sitting physically at the crossroad between York Avenue and 69th. which means you might hear any possible New York experience sound, like ambulances, people killing each other in the street. I mean, a lot of things going on across that window. So please don't be scared. I'm safe. I'm inside, but outside is kind of messy. And to share with you this picture, because in these times of pandemic, I always want to see the light at the end of the tunnel. And this is a picture I took myself in 2017 of a solar eclipse from Central Park. One of the best experience I had in New York at that time. Okay, let's have a look at first as a framework on how cells respond to stress. Okay, stress is placing cells at danger, not just cancer cells, but all cells in general. And cells rely on intracellular mechanisms of adaptation. Those mechanisms of adaptation can be successful, and in such case, cells recover homeostasis and physiological function, or they can be non-successful, and in such case, the cells undergo two of either terminal fate that are cellular senescence and regulated cell death. We will talk extensively about both later on. So if you think about a classical case for cancer therapy, when you use radiation or when you use some chemotherapeutics, you induce DNA double-strand breaks. And that is a sort of stress for the cells. And there is a machinery that detects those breaks. It doesn't matter really, here I put the proteins, but it doesn't matter that you recall the proteins, just a conceptual framework, right? That machinery, first of all, generates a pro-survival response. So first of all, the machinery of detection of stress will try to repair damage, will allow the cells with the possibility to repair damage. And this is, of course, in this case, is mediated mostly by autophagy, and we will discuss what autophagy is, and by a temporary cell cycle arrest, whereby the cells do not duplicate damaged DNA, but engage in repair. And again, if this is successful, the cells will survive. If this is not successful, the same proteins that mediated the pro-survival signals will convert into proteins that can mediate cell death promoting signal. And in this case, we're talking about p53. It's a very common oncosuppressor that when cells are damaged beyond recovery is able to activate mitochondrial permeabilization through BAX and other BH3 proteins that you probably have heard during the past sessions to, again, drive this regulated cell death. So what is regulated cell death? And this is a concept that I tried to introduce myself with Guido a few years ago to differentiate it from the previous wording of programmed cell death. Basically, cells can die in two diametrically opposed manners. One is an accidental manner, and one is a regulated manner. Accidental manner means when cells are exposed to such a harsh conditions that they basically disassemble immediately with no absolute control from mechanisms or proteins, so no genetic regulation, anything. Imagine the case that you have your cells in a plate and you burn them. You just place a fire on them. They will just break into pieces. They cannot do anything. Doesn't matter what proteins they express. Doesn't matter what cell cycle phase they're on. Doesn't matter what the medium composition is. They will just disassemble. On the contrary, when the stress is relatively milder, and so is the scenario I depicted in this slide, the cells will die because they activate a mechanism that is genetically encoded. So there are proteins and other factors in the cells that are there to kill the cell. And this means regulated, right? So that's a type of cell death that is controlled by a mechanism. A subsection of that type of cell death is programmed, and that refers, or at least we use it to refer to a cell death type that is completely physiological. There's no stress involved, but as you know, when your body develops during embryogenesis and also in adultism, homeostasis, there's a lot of cell death going on. And those cells are not stressed at all. They just need to die because of the ability of your organism to develop. And we call that programmed. But I want to really go to the basics here because I think it's important, especially for young audiences. You guys are, and I'm not talking about you, Gustavo. That is, when is a cell actually dead? Because it seems a very trivial questions, and it would be trivial if we could look under the microscope and see these kind of figures, but we cannot. So we have to take a very solid definition of cell death that will be operational and will allow us to really put a framework on what we do. And I had 2 videos here that will not work, so I'm going to skip on that. But the overall message of those videos is that cell death research is very messy, especially when it's done by non-cell death experts. So sometimes you get a very, very interesting Nature paper that starts with a figure 1A, and that figure 1A is based on a wrong method to detect cell death. And the entire foundation of the paper is virtually wrong, but nobody realizes because the paper has, for the rest, nothing to do with cell death. And so it doesn't go to cell death expert for reviews, and they will take whatever is in that panel for granted, because there's a lot of misconception around. Major example here, and we can discuss about in later on if you want. ATP, intracellular ATP as a manner to detect cell death, and everybody agrees that if you have 100% ATP levels in control cells and 5% levels in treated cells, this means 95 or all the cells were dead. It's a very extreme scenario. The point is happening when you have 80% residual ATP. And some people would interpret that as 20% of the cells are dead. It's not necessarily the case. It could also be that all of the cells have reduced their ATP levels to 80%. And so the reading that you get is going to be the same, but you will not be able to understand if the cells are dead or not unless you use a stronger, more direct type of measurement. So when is a cell death? What is the answer to the question? The cell is dead when the cell has irreparably, irremediately, irreversibly lost plasma membrane integrity. It's not when the cell, that is not when the cell is dead from a teleological perspective. Probably the cell was dead before that, but it's the only way that we have to be sure that it is irreversible. And many people, including my previous boss and many others, have tried to put an irreversibility point, the so-called point of no return, somewhere in between the activation of a stress response and this loss of plasma membrane integrity, but it is really complicated. It has been proposed caspase activation. Sure, caspase activation can drive cell death, but sometimes it does not. Mitochondrial permeabilization, yes, if enough mitochondria are permeabilized, probably the cells will die. But we don't know, in both these examples, we don't know at which degree the process becomes really irreversible. We know instead that when the plasma membrane is broken into pieces, then it's irreversible. So we use that as a very operational mechanism. So again, cells are alive and happy. You observe caspase activation, those cells can still be alive. You observe autophagy, and this is a worse scenario. We're going to discuss that later. Cells are alive, but they are dead when the plasma membrane is permeabilized. Also, perhaps in conjunction with caspase activation, also perhaps in conjunction with autophagy. But it doesn't mean because of caspase activation, doesn't mean because of autophagy. Here we are still talking about some correlations, and correlations are really dangerous in cell death research, really dangerous. And now I'm going to bring you 2 examples. We're going to show you a picture of my grandma and my ex-wife, and I'm sure they would both hate me if they knew, but since nobody knows, we're going to be fine, to tell you how important it is to have this strict definition of cell death. This is the case of my grandma. She's actually 95. She turned 95 3 days ago. And one would ask, potentially, if we were doing cell death research, if she's dead, right? Is my grandma dead? And one would ask you that because if you look, for instance, at one of the assays that radiation oncologists like so much, which is clonogenic assays, if you take my reproductive cells here, marked in A, you would see I can still generate colonies in a clonogenic assay, but she cannot, right? So if you go to our core radiation oncology definition, my grandma is clonogenically dead. And for whatever that means, right? But the problem is that does it mean she's really dead? It actually does not, because my grandma has a very strong secretory phenotype. She produces a lot of goods that then can be fed to who is not chronogenically dead, which is myself. So this goes to tell you one of the major wrong definitions that there are around cellular senescence. Cellular senescence is not a form of cell death. Senescent cells are alive, they are metabolically active, they have a secretory phenotype, and that can be very important in cancer therapy. Because for a long time, there was this idea that it's good if your cancer cells cannot proliferate anymore. Yeah, it's not bad. But if those cells, rather than being dead as we really wanted them to be, they are senescent, they will produce a lot of factors that will actually go to serve the metabolism of cells that are around, including cells, cancer cells that did not become senescent, and those will be provided with an advantage. So senescence is not a form of cell death. Let's clear that out. Second example, my ex-wife, I cannot tell her age because then she would really want to kill me. We were happy for a while, actually, but we divorced, and it was completely my fault, and now we're friends, and there's no problem about it, right? Why? What happened there? So there are several causes of divorce, right? And but if you look at the percentages of cases of divorce that are preceded by marriage, is 100%. Divorce is invariably preceded by marriage, as you obviously understand. So somebody can interpret that as in marriage causes divorce, which may or not be the case. When you think about regulated cell death, which is the topic, actual topic of this conversation, and not why I divorced from my ex-wife, 100% of cases of regulated cell death driven by stress are preceded by attempt of the cells to save their asses, to use a very French term. Cells want to survive, so they will mount an adaptation mechanism. Many people, interpret as if adaptation to stress causes cell death. And they invented, which is something I'm pretty skeptical about. And they invented this fantastic term called autophagic cell death, because they invented it at a time where the only tool they had was a microscope. So what they were doing, they were looking cells at the microscope, they were seeing this fantastic, nice autophagosome here eating cellular components, and then they were seeing the cells that were dead later on. But they couldn't prove that there was a mechanistic link. There was just a correlation. And indeed, we now know that autophagic cell death exists in some organisms, not very much so in mammalian organisms, but it's most of the time when we see autophagy is a cytoprotective mechanism that can be successful or not. If it's unsuccessful, the cells will die, but they will not die because of autophagy. They will die despite autophagy. And still, when you look at the literature, I mean, desperation comes out and all these sentences that seems to point to the fact that autophagy is a cause of cell death. And it's really painful for me to see that. So as I mentioned, autophagy, and we will discuss about autophagy extensively later, is a form of cytoprotection. There's many variants of autophagy. This is the most dramatic one. There was the most visually impactful one, the one that you see in that electron microscope picture, because it involves the formation of a new organelle, a double-membered organelle called autophagosome. Autophagosomes are there to pick up trash and potentially dangerous entities from the cytoplasm. For instance, invading bacteria or protein aggregate or permeabilized mitochondria. So how did we determine that autophagy is actually a cytoprotective mechanism? Of course, when the genetic era and the pharmacological tools became available, we knocked out genes that were involved in autophagy, or we used pharmacological inhibitors of autophagy. So we did a mechanistic experiments. And when you do that, your cells that are stressed by several stressors, autophagy is very important to compensate for multiple types of stress, they will die faster. They will die more. They will not die less. It means that autophagy is actually there to save the cells. And this is another example about how the dangers between the danger of focusing on correlation. Correlation, of course, we do observational experiments. Everybody does observational experiments, But one must be very cautious then in transforming this correlative data into mechanistic data. This is the number of people who drown by falling into a pool. Unfortunately for Nicolas Cage, it correlates with the numbers of films that Nicolas Cage appeared in, if you plot it over time. And I'm pretty sure everybody agrees that even though maybe Nicolas Cage is not the best actor on the planet, he's not responsible for those deaths. And this is, again, a funny collection of facts about water. And everybody agrees on those, and everybody agrees that water is not responsible for anything that is listed on this slide. So, and again, this is a very dear argument to me, a dear topic, because I happen to, be at the lead of a committee of people who actually care about the same things, and we reunite over the years to define conceptual, etymological, and mechanistic problems related to cell death and autophagy research, because we really want to see the field move forward, and there is a lot of confusion in the field and around the field. And this is the latest one. You can expect the 2021 version to come out soon. So let's go back a little bit to cell death in all its complex manifestations. So cell death manifests in multiple ways, right? Manifests in a morphological way, manifests in a biochemical way, manifests in a immunological way. But for a long time, people thought that these three aspects correlated. So thoughts that when you see cells that look like an apoptotic cells, you would also expect that cells to have massive caspase activation, and you would also expect that cells not to induce an immune response, because they thought like there was a dichotomy in cell types, apoptosis, this kind of cell death, that occurs with a set of manifestations that they always go together. It's like strawberries are always red. So if you have a strawberry, it has to be red. If you have a cells that look apoptotic, it has to be non-immunogenic. And we actually now know it's not the case. But all these definitions serve a purpose. And all those dimensions can be important and should be investigated. So the original definition was really a morphological one, which is now basically abandoned. I mean, there's still little people that want to engage into this morphological definition. And of course, why it has been abandoned, because here you see, classical morphology of apoptosis, blabbing, nuclear condensation. You can see also here, these areas of heterochromatin, very defined, very easy. I mean, I can give you all 25 pictures of this type, and you would all say, oh, this is an apoptotic cell. And this was the opposite version, which is necrosis from a morphological perspective, remember that. And it was ballooning, it was, enlarging, swelling, and then breakdown of the membrane. And again, if we give you 25, 50 pictures of those, you will be able to say it's necrotic. The problem comes with, these are too extreme of a spectrum. So when you really do a lot of VM on dying cells, you will see cells that look intermediate, cells that are swollen, but they have chromatin condensation, cells that are they don't have a precise phenotype. So how do we classify those? Is it going to be apoptosis or necrosis? Maybe I'm going to call it apoptosis, and some of you would call it necrosis. So that's where it has generated a degree of complexity, because it's not a dual scenario; it's a spectrum of phenotypes. Biochemical definition is actually way stronger. They are way less dependent on how I perceive it, because again, we have genetic tools and we have pharmacological tools. So if I measure cell death the way I told you, which is by measuring plasma membrane integrity, and this is very simple, especially in samples, because you use so-called vital dyes. Vital dyes, the most common one is probably propylium iodide, but there's a bunch of them. They get extruded only when the cells are functional and the membrane is intact. Otherwise, they can enter the cells. So you measure cell death that way, which is a very strong way. And then you measure cell death in the presence of a pharmacological or genetic inhibitor of your pathway of choice. For instance, if we think about like apoptosis now is currently defined biochemically as a caspase-dependent type of cell death. So What happens if you want to know what type of cell death you're looking at, you measure your cell death by PI uptake, and then you measure your cell death in the presence of a caspase inhibitor or upon the deletion of caspase 3. If there is less cell death, it means that that's an apoptotic cell death. As easy as that. And as strong as that, because you don't measure it by your eyes, you measure it by a number on a machine. And of course, we can debate how good the settings are, but it's way stronger than to look at the phenotype that can be misinterpreted. And again, here I just mentioned some of the major cell death pathways, but there's novel accumulating cell death pathways, because those definitions are actually based on the discovery of new players, molecular players in the cell death. And then there is, there is just a list that we did for the fun of the different cell death pathways. And you see, we moved from a situation where we had apoptosis and necrosis, which is basically 2005 or little more than that, to a situation when now we have at least 10 to 15 different cell death types when you define them biochemically. And then there's the most important component of the cell death pathway, at least for the purpose of this talk, which is the immunological dimension. So what happens when the cells die? For a long time, people investigated cell death in plates, acceptable. When they were doing cancer, they were also investigating cell death in an immunodeficient mouse that was xenografted with human cancer cells. And of course, it was important because those were the tools that are available, but it was completely ignoring the dimension, which is in normal human physiology, cells do not die in a plate and do not die in an immunodeficient setting. They die in the presence of an immune system that is there to decode the signals that the cells are providing when they die. And this applies to all pathologies where cell death is involved, either because there's too much cell death, ischemia, trauma, you name one, either because there's too little cell death, cancer. So it is very important, and it's what I'm investigating right now with my independent lab, to really think of cell death as something that happens in an environment that is not neutral. That environment will decode messages, and we're going to discuss about it later. But just to give you like an operational definition of what we now call immunogenic cell death, immunogenic cell death is a form of cell death that can activate an immune response that is specific for that cell antigen. Not inflammation, not general inflammation, no. Adaptive immunity specific for an antigen, which is different. And it involves several steps, and it involves several parameters. One, of course, is the identity of the cells that die. When the cells do not express antigens that can be recognized by the circulating TCR repertoire of a specific individual, they cannot be immunogenic because there's no T cells that can recognize those antigens. So even if at the very beginning we were focusing on immunogenic cell death as something that is most related to the death, we now move into a definition that involves the precise host where the cell death is happening. So that host must be able to recognize antigens that are released by those cells, must be able to decode these signals that are delivered by that cells, and must be able to locally mount an immune response, which sometimes is prevented, especially in the case of cancer. I know I have a cancer bias because I do cancer, but many of these principles apply to cell death driven by other causes, pathogen infection, in which case, for instance, the antigens are provided by the pathogen. So that is more immunogenic in the sense of antigens, right? But also other forms of antigens are being recognized. We have an interest in post-translational modification dependent antigens, and those are really a new form of antigens where you can lose tolerance to yourself, not because you express new proteins, but because your proteins are modified by oxidative and other modifications that can be enzymatic or non-enzymatic, but those modifications were not available in the thymus to shape your TCR repertoire. Before we really dig into the immunogenicity, two more general aspects which have changed over time. They have changed really a lot. For a long time, we thought that all these cell forms, like apoptosis, necroptosis, ferroptosis, they would be nearly linear pathways, meaning that when you were able to inhibit one of them, if that cell death pathway was involved in a pathogenic effect, then you would stop the pathogenic effect. Example, caspase activation. So if this panel A-B would be true, you would assume that blocking caspases with potent agents would actually prevent the death of cells, which in most cases is not the case. It just delays it, which is a different concept. And indeed, in the 90s, soon after the recognition that caspases were important for apoptotic cell death, the people developed caspase inhibitors, and they went to clinical trial because they wanted to save patients with ischemia or cardiovascular degeneration or aging when you have more cell death. And those drugs actually failed the clinic, with a marginal exception, which is Emricasat, which is now used. Well, it's not approved by FDA, but it's potentially usable for the treatment of cirrhosis, right? And this is not actually what happens. What happens is the cell death modalities are really interconnected to each other. So when you block one, most of the time you don't change the fact that the cells are dying. You just alter the way they will be dying if the initial stimulus is strong enough to drive cell death. Of course, if you block it ahead of time, you can really block it, but that's an upstream scenario that is not really common in human pathology. In human pathology, you intervene in people where cell death is already going on, so you would want to block the latest phase of the process, which seems to be complicated, at least. And then that is something I mentioned already. There are so-called essential versus accessory aspects of cell death. Again, when you have a perturbation of homeostasis, there is a adaptation to stress, and if it's successful, the cells will be happy and kicking. Sorry, this was not intended to happen. And if this adaptation to stress fails, then you go through this point of no return that is not particularly well defined. And then you have two processes. You have what really kills the cells. And if you block this, you will probably shift the mode of cell death toward another mode of cell death. But there's also a lot of things going on which are correlates. They're not mechanistically linked to cell death, but we can measure them. And that's important, that's been done. But we cannot really intervene on them from a pharmacological genetic perspective because we will not change anything about the cell death process. Those are just manifestation. And again, there is something going on beyond the death of the cells, which is what happens when the cells are dying in an organism. So both regulated and accidental cell death are associated with the release of molecule. Some of them are factors like reactive oxygen species, and others are called danger associated or damage associated molecular patterns, which are danger signals that can modulate the reaction of the body to dying cells, and they can modulate, and they will be very important for the ability of the body to take care of dying cells. Because at the end of the day, we move from being unicellular colonial organisms back in evolution, where the most important thing was that you as a cell wouldn't die, to being multicellular organism, highly evolved, in which the most important thing is the organism. And the organism knows very well that cell death is actually an homeostatic process. Cell death happens because cells are not functional anymore, because cells are potentially oncogenic, because cells are no longer needed. And in all those cases, cells are dying, and that's part of the way of the organisms to preserve homeostasis. And so the signals that are released by cells that are dying will determine whether such cell death will be pro-inflammatory, will be immunogenic, or will be tolerogenic, or at least will be ignored by the immune system, which is something I don't believe it happens, but some researchers want to say that some cell death can be ignored. I don't think there's any ignoring. I think there is a very much of an attention from the immune system to dying cells, and that is, it can lead to two completely diverse outcomes. It can be inflammation or adaptive immunity or tolerance. So what are these DAMPs or damage-associated molecular patterns? Those are the signals that the dying cells release that will talk to the immune system as if the interlocutor, as if the audience were a microbe. Those are endogenous molecules that are secluded within the cells, or they are existing in biochemical configuration that are incompatible with signaling. Example, intracellular ATP. Intracellular ATP in a cancer cell will not be detected by the immune system because it's inside the cancer cell. Intracellular HMGB1, so seclusion here is one of the major principles to regulate danger signaling. Those molecules are potentially inflammatory. They can boost an immune response, but they're not seen by the immune system in normal condition. They are inside the cells. When instead they are released, because the cells are dying, and they can be released even before the cells are really dead, they can be released by active phenomena that happens together with adaptation to stress, and we can discuss this later. So when those molecules are released, they become visible to the immune system, and the immune system has receptors that really interact with these molecules. And those receptors are called PRR, or pattern recognition receptors. The fun thing is, as I mentioned before, pattern recognition receptor did not evolve to detect endogenous DAMPs. They evolved to detect exogenous DAMPs provided by microbes that are called microbe-associated molecular patterns, or MAMP. Somebody called it pathogen-associated. I don't like pathogens because a totally non-pathogenic form of Escherichia coli will have LPS exactly as a pathogenic variant. And LPS is the molecule that would drive danger signal in that case. So pattern recognition receptors were there before we needed them for endogenous recognition. They were there to preserve us from invading pathogens, which is one of the first needs in evolution. And then they evolved also to be able to detect what happens inside our bodies, including all these DAMPs that I just mentioned. And ATP is one of the major DAMPs I'm going to discuss, but there are others. There's caroteculin. Caroteculin is an endoplasmic reticulum protein that is exposed on the membrane of cells that undergo immunogenic cell death. There's HMGB1. HMGB1 is a nuclear protein that is released when the cells are undergoing immunogenic cell death. And there are others, including mitochondrial DNA, which is also, we're going to discuss later. Mitochondrial DNA, as long as it's inside the mitochondria, there's no problem. When it goes to the cytosols and when it goes to the extracellular environment, then it operates as a DAMP as well. So why are DAMPs so important for cancer therapy and for actually the detection and the recognition of immunogenic cell death? Because they are needed for the immune system to interpret the situation of dying cells that are immunogenic. This is a paper that was published back in the days, back before I joined Guido's lab. We talk about 2005. And these guys were smart enough to discover that when you use specific chemotherapeutics in vitro, and you kill cells with those chemotherapeutics, and then you remove the chemotherapeutics from the cell culture, and you use dying cells as a vaccine, you can protect mice from the development of a second injection of the same cell type. And here you see it on the left. Here, these were the preliminary experiment with doxorubicin. Doxorubicin is an anthracycline that is used in the treatment of breast cancer and other forms of tumor. When the mice were vaccinated with PBS, of course, and then 2 weeks later, they were challenged with CT26 cells in this case, most of the mice developed a tumor. But when they were vaccinated with dying cells, you had 40% or 30% of the mice that were protected from tumor development after the injection of another lesion. So basically, dying cells by themselves were able to generate a vaccine type of protection. And those were really preliminary experiments where these guys were getting 40% of protection. Lately, we were getting 80, 90, even 100% of protection when we refined our protocols. So dying cells can initiate this adaptive immune response. And this is specific for the cells because when you challenge those protected mice with other cells, they are not protected anymore. So if you challenge them with TSA, which is a breast cancer cell line, they all develop a tumor. Doesn't matter whether they were previously vaccinated with CT26 cells. And instead, when you re-inject them again for the third time with CT26 cells, those mice that did not develop a tumor here cannot take even that second injection. And we spent a lot of time trying to understand which molecules were important for that effect. And we realized pretty soon that the release of extracellular ATP was important. Here you see what happens when you treat mice that are bearing a tumor that is engineered to detect extracellular ATP. So we engineer CT26 cells to express an extracellular version of luciferase that is activated by ATP. So when we treated these tumors that were control tumor with mitoxantrone, which is another anthracycline that induces immunogenic cell death, we would see an increase in extracellular ATP signaling, and that increase would be prevented when we used ATG5 or ATG7 knockout cells, so cells that are unable to undergo autophagy. You see here, we wouldn't see any signal from that. This is still a correlative experiment, so we observed then when autophagy is blocked, these cells are unable to secrete ATP. But what happened to the vaccination effect? When we tested those cells for the capacity to vaccinate mice in the context of immunogenic cell death, so when we use these cells as a vaccine, when the cells were wild type or they were treated with a controlled small interferon RNA, and then with mitoxantrone, we got this very nice protection, which we lost when the cells were unable to undergo autophagy, so when the cells were unable to secrete ATP. So in this case, immunogenic cell death happens only when these damps, specifically ATP, can be properly emitted by the cells and can be properly detected by the environment. This is another example. Probably here you will see the movies. These are wild-type cells, wild-type cancer cells here that are dying in response to immunogenic cell death. And the little things that you see passing by are splenocytes, so are basically immune cells. And when everything is wild-type and you kill the cells in the right way, as we did, you see here that there's a good targeting. So the splenocytes are actually targeting the dead cells. And you can quantify that in interaction time here. When those splenocytes are missing one receptor that is called FPR1, which is a receptor for annexin A1, which is another one of those danger signals, there is no targeting. The splenocytes are ignoring the dead cells here. They're just passing around, and when you look at the interaction time, you quantify here, is basically as if the cells were not dying, or if the cancer cells were missing this danger signal. So you need a pattern recognition receptor on the side of the host and a danger signal on the side of the cancer cell for this to happen. And when you look at treatment efficacy in this case, so when you have wild-type tumors and wild-type mice, you have a nice effect. When your tumors are lacking the danger signal, the effect of mitoxantrone is virtually gone, as it is gone when the tumors are where the mice are lacking the pattern recognition receptor, in this case, FPR1. So you really need a couple to work, something going on from the cancer side, from the dying cell side, and something to operate in the host. And when we instead delete the pattern recognition receptor from the cells, nothing happened, because again, the pattern recognition receptor has to operate on immune cells, not on cancer cells. So this is immunogenic cell death, as I mentioned, and it starts way before the cells are really dead. It starts when the cells are answering and responding to stress. As I mentioned, ATP secretion, which is the ultimate danger signaling going on here, depends on autophagy induction, and autophagy induction starts before the cells are dying. The exposure of endoplasmic reticular protein on the membrane of dying cells, including reticulum, as I mentioned, starts when the cells are undergoing ER stress. They're not dead yet. They are responding to stress. And the same happens for other things, including type 1 interferon secretion. This starts in the cells when they undergo a viral response. So they activate another pattern recognition receptor, which is called TLR3 or cGAS, which we'll discuss later on. So how do you identify immunogenic cell death inducers? And this is important. You really want to focus on dying cells here. You don't want to focus on the potential of other effects. So you have two things to do. One is tumor growth experiments, which are not definitive. and one is vaccination experiments, which are definitive but very sensitive, so we have to take it with caution. Tumor growth experiments. So the idea is cancer cells are dying and they activate an immune response, and it is part of the efficacy of the treatment. So obviously, in this case, you will only get an optimal treatment response when you use immunocompetent mice, because when you use immunodeficient mice, there is nothing going on from the part of the mouse. So yeah, chemotherapy will kill some cells, but not enough. And the immune system will not kick in because those mice don't have an immune system. So you will get in tumor growth experiments, you will get only an optimal response in immunocompetent animals. But this is also the case when you use drugs that are immunostimulatory for other reasons. For instance, some chemotherapeutics not only kill cancer cells, they also kill immunosuppressive cells from the tumor microenvironment. So in the very same way, they boost anti-cancer immunity. Well, they different mechanisms, but they also boost anti-cancer immunity. They don't induce immunogenic cell death, but they promote the ability of the host to mount an immune response. So you will have the same thing here in the absence of pure immunogenic cell death. When you want to study pure immunogenic cell death, you have to do vaccination experiments. So you kill your cells in a plate, in a dish, with whatever treatment you have in mind, and you want to know if it's immunogenic or not. For instance, radiation or chemotherapy, doesn't matter. So then you have a mix of dying cells that you clean from the chemotherapy because you don't want to have the effect of the chemotherapy on board. You just inject dying cells. And then you come back 1 week later and you inject living cells of the same type. So if the mice are protected, as in a vaccination experiment, prophylactic vaccination experiment, then it means that those dying cells, the only thing that you injected, were immunogenic enough to mount an immune response. If they were not, of course, the mouse will develop a tumor. What's the problem here? The problem is that cancer cells tend to be immunogenic anyway, because they express antigens that have evaded immune recognition. So they are this is a very sensitive assay. The mouse didn't see a cancer before, you get them a cancer. So if you get some danger signaling, that's fine, it's gonna work. But this is not what happens in humans. So in humans, there's a lesion that is there and it's already immunosuppressive, which is difficult to target. And this is so true that for some cell type, you don't need to have dying cells. For instance, CD26 cells, you can inject them, they will form a tumor. If you do a surgery and you remove the tumor, and then you try to inject them again, the cells, they will not grow. Because even if the original cells were not massively dying, the mouse was able to mount an immune response anyway. Of course, was not good enough to protect the mouse from the initial challenge because the cells were already replicating, but it was good enough to protect the mouse from the subsequent challenge in the absence of cell death. So this is to tell you how sensitive is this system. It's actually more complicated to see the second scenario as compared to the first scenario. And of course, here I said immunocompetent, but it's not enough. They have to be immunocompetent and syngenic animals. So the background of the mouse has to match the background of the tumor for obvious reasons. So another way to detect this, which is a little bit more clinically relevant, but it's actually very complicated in the sense that it's very hard to see immunogenic cell death in this setting, is what we call a Scopal experiment. And it's very relevant for radiation, and the limitation being that you have to have some sort of focal treatment going on. In this case, what you do, you put a mouse, immunocompetent, syngenic, with two tumors, one on one flank and another one on another flank. These two tumors will grow because the mouse has never seen them, and they will generate an immunosuppressive microenvironment that enables their growth. As in the human disease, tumors grow because they evade immunity, they generate immunosuppression. That's exactly the same thing's gonna happen. What you can do here is, if you have a focal treatment like radiation therapy, you can treat one lesion only. And if you treat that lesion only with a very powerful inducer of immunogenic cell death, like radiation can be in specific settings, the immune response that you will generate is so strong that it will go systemically and it will take care of the other tumor that you never treated. And this is called an abscopal response. And we observe it. If you radiate it, this is the down quantification of the tumor growth for the primary tumor, the one that potentially receives radiation, and the non-irradiated tumor. if you use a regimen where you have radiation plus an immune checkpoint blocker that is systemically inactive, and this is very important because if it was systemically active by itself, of course, you're not measuring an immune response, you're measuring the activity of something going on systemically. But in this case, a CTLA-4 blocker is not systemically active. So when you combine it with radiation, only in that specific setting, the non-irradiated tumor will be controlled, meaning that you generated an immune response such strong in this tumor that it goes systemically and it takes care of the non-irradiated lesion. Why is this clinically relevant? Well, there are several ways to prove it. And one way that we did, and we looked for polymorphisms in genes that encode such pattern recognition receptors in patients. And those genes are polymorphic. So for instance, the receptor for extracellular ATP comes in two variants, one which is a little bit less active. And when patients have this little bit less active variant, their metastasis-free survival upon treatment with anthracyclines, which is, again, a good immunogenic sedative use, is reduced, meaning that there is an impact from the host configuration. And again, in this case, this pattern recognition receptor. And basically the same thing happens when these patients express a slightly inefficient variant of TLR4, which is the receptor for extracellular HMGB1. So from a cancer treatment perspective, what you want to do is think about scenarios where you don't have good immunogenic cell death, either because the cells are not able to release those DAMPs, because they don't engage in a strong stress response, or because they are lacking the DAMPs entirely. There are HMGB1 negative cancer cells, for instance, or because they are unable to engage in pattern recognition receptor signaling. And we provided preclinical data in support of all these examples. For instance, when you think about ATP here, ATP is released because of autophagy. So if you are in a situation in which you have poor ATP release, maybe you want to combine your treatment with autophagy inducers, so they're going to have stronger ATP release. Or you want to inhibit the degradation of extracellular ATP. There's a lot of ATPases in the microenvironment. If you inhibit them, you're going to boost the level of extracellular ATP. Same things goes for caroteculin. If caroteculin exposure is not that good, maybe you want to use an ER stress to boost it, or you want to provide recombinant caroteculin to engage in the signaling that caroteculin would normally do. And the other examples go along the same lines. And is this clinically relevant is this relevant, this complementation concept from a clinical perspective? And we think the answer is yes. And to prove it, we went back to IGR and we picked up a cohort of patients that were so lucky to have both cancer and a heart condition. A heart condition for which they would be treated by the clinical agent. Now, I don't think it's ever used again, but in the past it was called digoxin. Digoxin is a cardiac glycoside, and we had demonstrated that cardiac glycosides are very good at boosting danger signaling, very good at promoting caroteculin exposure, ATP release, you name it. So if you think, and we matched that cohort to a cohort of patients who were only unlucky enough to just have cancer. One to two match cohort. So we try to make it very match in terms of clinical parameters. So they were matching sex, they were matching, stage of the disease, type of the disease, treatment they got. When you think about these two cohorts of patients, so one has just cancer and the other one has cancer plus a heart disease, for which they are treated with digoxin, you would expect that the cohort of patients with two conditions would be at a disadvantage in terms of overall survival. They would die before because they don't just have cancer, they also have a heart condition. And we found out it was the other way around. We found out retrospectively, with all the limitations that apply here, that the patient who had cancer as well as a heart condition treated with digoxin were dying slower. So we did one analysis further, and we stratified this patient based on if they were getting an immune checkpoint, an immunogenic cell death inducer or not as part of their treatment. And funny enough, even though these are very small numbers, very retrospective data, very cautious to be interpreted, but at least it supported the idea that when these patients, for instance, focus on the colorectal carcinoma scenario, when these patients were getting oxaliplatin, which is an immunogenic cell death inducer, it wouldn't really matter if they had an heart disease and also were getting digoxin or just cancer. But it mattered a lot when they were not getting an immunogenic cell death inducers, supporting the idea that the presence of digoxins was boosting the immunogenicity of cell death driven by treatment in that case. So let's go back. I think I have still some time, right, guys? I mean, in case I'm running out of time, shoot me, tell me. Going back to the general organization of stress response, I told you cells are having this thing. So, and they actually do what we do when we get stressed, like when we receive this message from our girlfriend, wife, or you name it, we panic and we need to adapt to that. And if you're lucky enough, we will be provided by material and support to recover homeostasis. And in that case, it's a drink, at least for me. And if you think about autophagy, as I mentioned before, autophagy sits exactly here. Autophagy sits as a way of cancer cell to cope with stress. So it was not a particularly smart hypothesis to think that if you want a cancer cells to die, you should inhibit autophagy, right? And this is a very standard approach that has been undertaken to combine autophagy inhibitors, that some of them are actually clinically available, and there's a lot of discussion how good they are. They're actually very bad. But it was worth giving a shot to the idea that when you kill cancer cells with any form of treatment like radiation here, you generate a quantity of cell death. But cancers are very heterogeneous, so not all of the cells die, because if all of the cells died, we would have beaten cancer long ago. Doesn't happen. You treat them with radiation, some of them will die, some of them will not die. But what we can do is, at least from a theoretical perspective, use autophagy inhibitors to maximize or to boost that cytotoxic response in terms of quantity. And I like to call this approach the Starbucks approach. And actually, the Starbucks approach works pretty well, especially or mostly if you do it in immunodeficient mice, which is a list here, that you use as host for human cancer cell lines. So in that case, it works. I mean, there's a lot of literature, even in place, it works well. And this is an example from our lab, from Guido's lab in the past. We had control cells scrambled, so this circle, or autophagy incompetent cells in these squares that would get or not get radiation. So if you irradiate them, they respond. If they are in an immunodeficient animal, they respond to some degree because radiation is a cytotoxic agent. So if you also inhibit autophagy by genetic means, they actually respond better, not surprising. And one would conclude from that the Starbucks approach worked, which is why people went then to the clinic and they give the only two drugs that can be used to block autophagy, which is chloroquine and hydroxychloroquine. That you probably heard about them now in the COVID pandemic for the wrong reasons. Those two drugs have been used in the past for treating malaria. They are lysosomal inhibitors, so they are very non-specific. They inhibit anything that goes through the lysosome. But again, autophagy goes to the lysosome, so they also inhibit autophagy. And people combine them with a lot of treatments just to see whether they would have some clinical advantage in the context of cancer treatment. And the answer is no. All these clinical trials and others to follow this relatively outdated scenario were basically negative. I mean, yeah, the safety of the drugs was not particularly concerning. They are not very safe, but there are side effects. But I mean, in cancer patients, those are manageable. But there was no additional efficacy on top of what you would expect from the standard care. Why? That we thought, because of the reason I mentioned before. I mean, cancer cells don't die in a plate. They don't die in an immunodeficient animal. They die in a very immunocompetent patient. I mean, very few cancer patients are actually immunodeficient. They can have some neutropenia because of treatment. They can have some myelosuppression, but most of them can mount normal immune responses to vaccination. So they are just immunocompetent. So we thought the problem would be when we inhibit autophagy, so we inhibit this thing here, we block danger signaling, so we inhibit the involvement of the host in the potential efficacy of treatment. So we proposed an alternative approach. Why don't we use instead autophagy activators, we know that we're going to give away part of the cytotoxic response. So we know we're going to kill less cancer cells, but maybe we're going to kill them with a better quality. We're going to kill them with a quality that enable an immune control of the disease. And then if the immune system jumps in, the heterogeneity of the cancer is less important. Not completely non-important, but less important. And I like to call this approach the espresso approach because it talks about quality of cell death, not quantity of cell death. And I mentioned this before, there are several examples where inhibiting autophagy is detrimental in the context of an immunocompetent host. This is the same experiment I mentioned, the vaccination with autophagy deficient cells that are treated with an immunogenic cell death induces in vitro doesn't protect the animal. So it means something is going on. If you go to a treatment setting, same cells growing in a wild type animal treated with mitoxantrone, the efficacy of mitoxantrone goes away when these cells are autophagy deficient, really compromised 100%. If you repeat the experiment I mentioned before, in an immunocompetent host, so colorectal carcinoma cells, autophagy knockdown, in this case, the efficacy is reversed. So the best efficacy, you get it when you irradiate control cells, not when you irradiate ATG knockout cells, which is the contrary of what I mentioned before, because here the immune system is more important. The contribution of the immune system is the dominant one to the efficacy of the treatment. In this case, there's no immune system. Of course, there's no contribution from it. Of course, we wanted to go one step further, which is we had demonstration that blocking autophagy can be detrimental, but we didn't have any demonstration that boosting autophagy is beneficial. And the best way to boost autophagy, very safe way, is to use caloric restriction. Autophagy has a major nutritional role. So when you deprive cells or mice or people from food, they will mount autophagic responses. And in this case, we combine that cycle of starvation, which is a weekly cycle of starvation, with chemotherapy. Again, immunogenic chemotherapy, mitoxantrone or oxaliplatin, immunocompetent animals. And this is a model of fibrosarcoma. You see it very well here. You see it very well here. When you use immunogenic chemotherapy alone, you have an effect. When you combine it with starvation, you have a better effect. So boosting autophagy in this animal provided an advantage in terms of tumor control by immunogenic chemotherapy. And it was actually due to the immune system, because when we did the same experiments in immunodeficient animals, everything went away. No more efficacy from mitoxantrone, no more impact from starvation. Of course, when you talk about starvation, clinicians don't like it. They have a point. Starvation in cancer patients, even cycles of starvation is complicated. So we move very quickly toward molecules that we call caloric restriction mimetics, because they have the same biochemical effects of starvation. Notably, they are very potent activator of autophagy, but they don't engage any weight loss. This is what happened when you starve a mouse for 24 hours. The mouse loses 10% of the weight. If you starve a mouse for 48 hours, which is the upper ethical limit to do it, they lose 20% of the weight. If you think about that in human terms, a person like me with 75 or 70 kilos would mean losing in 2 days 14 kilos. Okay, of course, it doesn't happen in humans that way because mice are different animals, they have different metabolism, but to tell you how potent that is. When you treat these animals with the caloric restriction mimetics, including hydroxycited spermidine, rapamycin, resveratrol, they have very potent activation of autophagy, which you see here in the Western blot, but they don't lose weight. So it's actually better. And those molecules behave like starvation also in terms of promoting tumor control by chemotherapy. And you can see here, all the red curves are better than the blue curves. Each of the plot is a different caloric restriction mimetic. And in all cases, it goes away because when you use immunodeficient animals. So basically, we demonstrate here that activating autophagy with caloric restriction mimetic or caloric restriction boosts the immunological control of the disease by chemotherapy. We had the same results in the different model. This is a model of hormone receptor-driven breast cancer that it's very dear to me. And you see here the survival extension and the improved tumor control when you combine mitoxantrone and hydroxycited caloric restriction mimetic. And it's compromised when you block CD8 positive T cells. You see here, you go back to control settings, also in terms of survival. So second round of conclusion. the espresso approach is better. And actually, when I moved to New York, I was really convinced that the espresso approach is better. Unfortunately, as many times in life, you have to consider things from a better perspective. So should we inhibit or activate autophagy? And it's not very simple, because I mentioned autophagy is very important for the cells to survive stress. So you may wanna, if you think about cancer, you may wanna inhibit autophagy, but it's also very important for the cells to deliver signals to the immune system, both locally and systemically. So again, if you think about cancer cell, you may wanna promote autophagy. And this is actually changing a lot depending on where you look at. And this is what I started doing when I moved to Cornell. And then, so I moved to a radiation oncology department. I mentioned the abscopal response before. The abscopal response is this magic phenomenon whereby you irradiate one tumor and many others go away. This is an example of a non-small cell lung carcinoma patient receiving irradiation to an hepatic tumor, and a few months after, all the systemic disease was gone. Very rare clinical event, became more frequent with the introduction of immune checkpoint blockers because it's an immunological event. And when you move to a radiation oncology department, it's the holy grail. It's what you want to understand, it's what you want to find. So we also started working on that. And there was data from previous work in the department demonstrating that this holy grail comes from the ability of radiation to generate the accumulation of cytosolic DNA. So when you radiate cells, they accumulate DNA in the cytosols. That drives the secretion of type I interferons through a mechanism that depends on a cytosolic sensor, which is called cGAS. And that recruits dendritic cells. Dendritic cells migrate to lymph nodes, and they generate an immune response that can go and take care of the cells that didn't die because of radiation, but can also go and take care of the abscopal, so of distant tumors that were not irradiated. And this is really another way of calling immunogenic cell death. You kill these cells in a way that they mount a strong immune response systemically. And there was a lot of literature available when I moved in, demonstrating that autophagy inhibits type I interferon secretion. So at least theoretically, it should inhibit the abscopal response. And so we started investigating the role of autophagy in abscopal responses to radiation. This is work from one of my postdocs, Takahiro, and a fellow that was in the lab for a few months, Marissa. So to do that, we generated breast cancer mouse cell lines. So it's called TSA cells, I mentioned them before, that are deficient for ATG5 or ATG7. ATG5 deficient cells, of course, lack both the conjugate version of ATG5 and the monomeric version of ATG5. The cells that are deficient for ATG7, they lack ATG7, as you can see here, and they also lack the conjugated version of ATG5 because ATG7 is the enzyme that conjugates ATG5. We checked that these cells were actually autophagy deficient in both starvation and radiation condition. This is a standard way to measure autophagy. You measure whether the autophagic substrate P62 gets degraded and whether this is dependent on lysosome. This is starvation medium, and indeed, if you compare this lane to this lane, you see there is a degradation that is prevented by a lysosomal inhibitor. So this is bona fide autophagic flux, which you can also measure by monitoring the lipidation of LC3. Radiation does the same trick, so radiation activates autophagy, and this is, again, prevented by a lysosomal inhibitor, and of course, it does lipidate LC3. And this does not happen, as expected, when the cells are ATG5 or ATG7 deficient. There is no degradation, and there's no lipidation at all. So we were basically validating the model. We tested whether these cells would be more sensitive to radiation in vitro. As I mentioned, autophagy is a cytoprotective mechanism. We would expect them to be more sensitive. And indeed, when we measured PI negative cells, so living cells, irradiated them with different doses of radiation, and the ATG5 in blue or ATG7 in pink deficient cells were more sensitive. Nothing particularly spectacular. This is when we started to have issues, or I started to have personal issues. Again, you're seeing the same experiment before. ATG5, ATG7 knockdown cells were unable to protect mice from a challenge with living cells in the context of vaccination. But when you use radiation in vitro to kill the cells, then it no longer matters. You see here, this is the ability of dying cells to protect the mice from an injection with living cells. If you use control cells, 100% of the mice are protected. If you use ATG5 knockout cells, still 100%. If you use ATG7 knockout cell, still 100%, which was really disappointing to me because I wanted to prove what we proved with chemotherapy in the past, so that it's important to have ATP release from radiation as a matter of immunogenicity. That no, doesn't matter. And indeed, when you go in immunocompetent animals and you treat these tumors with radiation alone, you get an effect. When you treat them with radiation plus an autophagy inhibitor, you get a better effect. Same thing when you treat control tumors with radiation or you treat autophagy deficient tumors. So it really seems that even in immunocompetent settings, autophagy is bad for the ability of radiation to mount an immune response. And of course, we were interested in type I interferon because of the Scopal response. So we started to measure type I interferon secretion either at the transcription level by RT-PCR or ELISA-based, in the medium. And we realized very quickly that when you inhibit autophagy in the context of radiation, you get improved type I interferon transcription. Same with autophagy deficient cells. Same when you measure the actual secretion of type I interferon in the medium. And this is not surprising again, because we knew from the literature that there is a type I interferon setting with autophagy. We made sure this was a cGAS and STING dependent effect because we knew that cGAS and STING are important for the ability of radiation to mount an abscopal response. And indeed, the capacity of radiation and the capacity of autophagy inhibitors to boost that effect is lost when you have cells that express cGAS and STING or when you have cells that you are treating with a cGAS inhibitor. So it's really a cGAS dependent effect. And it is a cytosolic DNA accumulation dependent effect. Again, we suspected it based on the ability of autophagy to pick up sources of cytosolic DNA, and based on the data linking type I interferon driven by radiation to that. And you can see here, this is a quantification. We developed an immunofluorescence-based protocol for quantifying cytosolic DNA specifically based on the selective permeabilization of the plasma membrane, but not the nuclear membrane. As you see here, we have a double-stranded DNA-specific antibody that does not recognize the nucleus because the nucleus does not get permeabilized. And this is an automated image analysis demonstrating that pharmacological or genetic inhibition of autophagy boosts the ability of radiation to drive cytosolic DNA accumulation. And at that time, there was a focus on micronuclei as the source of cytosolic DNA driving this process. And micronuclei are small nuclear fragments that accumulate when cells undergo faulty mitosis. And also Guido had published in the past that autophagy is very good at picking up micronuclei. So we really wanted to link all the pathway with micronuclei as well. It turned out that actually, when we stained the cells for this double-stranded DNA upon radiation, we were unable to colocalize double-stranded DNA to any nuclear markers, including lamin B, which is a marker of nuclei and macronuclei. So not happening here. Instead, when we did the same trick with COX-4 being a marker of the mitochondria, we noted that the double-stranded DNA species we were detecting in the cytosol were really close to mitochondria. And so we started to think there is a mitochondrial DNA implication here. So we quantified that. Those were movies. We quantified that. So you see the dots that colocalize with lamin B are very few, and instead they mostly colocalize with COX-4 structures. And they also colocalize with a mitochondrial transcriptional factor, which is called TFAM. So when you relate the cells, whatever accumulates in the cytosol is mostly, in most cases, also stained by TFAM, which is this mitochondrial transcription factor. And this is the quantification of it. So we had observational data. We wanted to have mechanistic data because, again, observation is important, but we need to find out mechanism in science. So we generated cells that lack mitochondrial DNA. And this is a simple procedure. You culture yourself for a long time in ethidium bromide. You provide them with a lot of supplements, and you generate cells that are highly depleted in mitochondrial DNA. You can see here, there's a PCR-dependent quantification of genomic versus mitochondrial DNA. And when you irradiate those cells, they do not accumulate DNA in the cytosol. You see here, you see the quantification. You can do it in image-based. You can do it by RT-PCR upon subcellular fractionation. So mitochondrial DNA, when it's not there, prevents the ability of irradiation to drive the accumulation of cytosolic DNA species. And this is same thing, we can skip on that. But more importantly, when mitochondrial DNA is not around, the cells are unable to respond to radiation by secreting type I interferon, which is at the basis of our findings. And we were happy to see that other people had similar results. These guys were actually studying mitochondrial DNA release by BH3 mimetics, and they had a very similar configuration that we did, in which mitochondrial DNA was actually bulging from mitochondria rather than being free in the cytosol. So we wanted to prove the implication of the apoptotic machinery in this finding. So we used the cells that overexpress BCL2, or they are knockout for BAX. As you know, BCL2 is a mitochondrial protector. It prevents mitochondrial permeabilization. BAX is a mitochondrial permeabilizer. And in line with that, when we had cells that overexpress BCL2, the amount of DNA in the cytosol was reduced and type I interferon secretion was reduced. When we had, and the same happened when we have BAX knockout cells, so less cytosolic DNA and less type I interferon secretion. So we really had a pathway going from mitochondrial DNA through BAX-dependent BCL2 control pores through SIGA signaling and type I interferon. So at this time we had two stories. One was the mitochondria story, one that was the autophagy story. We wanted to be sure that the autophagy story was linked to mitochondria, so that autophagy was actually picking up mitochondria and deciding, permeabilize mitochondria deciding. So to do that, we generate mtDNA depleted cells, so the Rho0 cells in the ATG5 and the ATG7 knock backgrounds. And you see here in control condition, you have an increase of cytosolic DNA accumulation when you radiate ATG5 knockout or ATG7 knockout. We saw this before. And this goes away when mitochondrial DNA is no longer on board. So autophagy here is really picking up permeabilized mitochondria. And the same happens when you measure type I interferon secretion. Again, it's higher in the ATG7 knockout, but it goes down to control when the ATG7 knockout is also lacking mitochondria. Or you can do the trick with pharmacological inhibition. Again, Rho0 cells don't produce type I interferon in response to radiation. We tested that as an abscopal response. I explained this in the before. So we have two lesions, we irradiate just one, and we check what happens in the non-irradiated lesion. But the first lesion can be wild type, or it can be ATG5 or ATG7 knockout. This is what happened in control condition. So all the tumors are control tumors, both the irradiated tumors and the non-irradiated tumors. Of course, the irradiated tumors, when it gets irradiated, it responds very well. It responds even better when there is a checkpoint blocker in the systemic checkpoint blockade, but the systemic checkpoint blocker alone doesn't do anything. So this is not important, because what we are interested into is the second lesion, so the lesion that never sees anything, never sees radiation, and the lesion for which systemic checkpoint blocker is per se inactive. But when you have radiation of the other lesion in the context of checkpoint blocker, then you can get a control of this lesion, and which is better with this 3 times 8 gray regimen for several reasons we don't want to go into now. But you see, we can get a solid control of the non-irradiated lesion. But what this tells you is how frequent we can eradicate the non-irradiated lesion. And the answer is, when you use wild type cells in the first lesion, so in the lesion that you do irradiate, you never get an eradication of the second lesion. But instead, when the lesion you do irradiate is autophagy deficient, sometimes you even get an eradication of the second lesion. And when you take these animals, which are now tumor-free because you eradicated both the primary lesion that was irradiated and the non-irradiated lesion, so like metastasis, when you get these mice and you re-challenge them again with those cells, they don't allow them to grow. And this is dependent on type I interferon, because when you use a type I interferon blocking antibody, the control here in blue of the non-irradiated lesion goes away. There is some translational value to that, and to check for that, we went into public data sets and we checked a gene signature that is representative of our system here. This is where ATG5 or ATG7 collaborates with each other. So we picked a signature including 5, 7, 10, 12, and 16, and 1, so 5 genes that we thought was relatively centered around our own work. And not surprisingly, patients with breast cancer that have a high autophagic signature in their tumor, they are doing worse in terms of survival as compared to patients who have less signature. And this is, again, we knew autophagy is good for cancer cells, so it's actually not surprising at all. But we were not really interested in that. We were interested in how this relates to anti-cancer immunity. And to do that, we checked gene signature for type I interferon signaling. As I told you, autophagy prevents type I interferon secretion in irradiated cells. And here, no matter whether you stratify your cohort in like median, tertile, quartile, the higher the autophagy score, which is on the x-axis, so this is high autophagy, this is low autophagy, higher autophagy, lower autophagy, the lower the type I interferon signaling you get. So it goes down here, it goes down here, it goes down here, meaning, again, high autophagy, low type I interferon signaling in the tumor of patients. And the same happens if you use a score for interferon gamma signaling, which is an indication of ongoing anti-cancer immunity. The higher the autophagic score, the lower interferon gamma signaling. And what we want to do was also to put it in the context of mitochondrial DNA in some way. Yeah, this is just a gene expression, doesn't matter. It goes again to say that high autophagy means low interferon gamma, even when you compare the transcriptional signature of that and you do classical gene ontology of hallmark-based enrichment analysis. We wanted to dig, I told you, on the mitochondrial DNA aspect, but of course, we didn't know how to do it because these data were collected in the pre-RNA-seq era. So most of them are actually microchip, microarray based data, where mitochondrial RNAs are not included. But we reasoned that the abundance of mitochondria would actually influence the abundance of nuclear genes coding for mitochondrial proteins. Because as you know, the more mitochondria you have, the more the nucleus has to provide material for the mitochondria to build. Because the mitochondrial DNA is only codes for only a few polypeptides, and it's not enough for mitochondria to be built. So we generated a score encompassing all nuclear genes coding for mitochondrial RNAs. Sorry, coding for nuclear RNAs that would code for mitochondrial proteins. And if you do the same trick, so if you separate your patients based on autophagy score, again, the higher autophagy you have, the lower of that signature of mitochondrial proteins you have. So should we inhibit or activate autophagy? There's a long story to be told about that. And now I'm convinced that it really is important to situate yourself precisely in terms of cancer type, disease type. Because if you are in this scenario, you really don't want to get an espresso, really advise against it. And if you are in this scenario, you will never get a Starbucks. Well, now I learned that the Starbucks opened in Milan, but it's for a long time, there was no Starbucks in Italy. And of course, the problem with autophagy remains that the current tools to modulate the system are very non-specific. First of all, they are very non-specific because most of them just target the lysosome. So they target anything that looks like a lysosomal process: phagocytosis, endocytosis, vesicular trafficking, autophagy, you name one. But also, we will want to have to develop targeted therapies for cell type. I just demonstrated to you that inhibiting autophagy in the cancer cells is good when you want to think about combining radiation. But we know inhibiting autophagy systemically is not good. Your immune cells need autophagy to mount an immune response. So when you use all these systemic autophagy inhibitors, you inhibit autophagy across the board, and that's not what you want to do. Yeah, we can skip on that. So just, I think the time is good to closing this up. This is my team now. I have 4 very talented postdoc and a technician and a golden retriever who unfortunately is not allowed to be in the lab, but provides a lot of support, psychological support to his owner. The data I showed you was completely generated at Cornell with the help of 2 great teams from Austria. Trojanowski lab did all the bioinformatics studies I mentioned in silico. The Garcia Martinez lab provided sample for studying mtDNA in patient. I didn't show you the data, but we had also a signature in patient. And of course, this is the radiation oncology department, and the people who are so crazy as to give me money to do stuff. And I think I bored you enough with all this story, and I'm happy to take any question you might have.
Thank you, Lorenzo, so much. It was a wonderful lecture. I think you did a great job starting from the very basics, even passing through some of the things that we find very important for the students, which is the technical approaches and so on. We have discussed that before, remember? And then now with very detailed experiments on this important immunogenic cell death and the role of autophagy and so on. I have a couple of questions myself, but I think we should start with the students. We have some questions at the YouTube, but
I have a couple of questions also.
I'd like to ask if you guys so maybe we start with the YouTube. If you or Ricardo can.
Okay, I can read the first one.
I think Karina wants to start with her personal question. Is that right, Karina?
Yes.
Okay, go ahead then. Ladies first.
Thank you, Gu. So I have a couple of questions. Since the crosstalk between autophagy and inflammasomes is extensively reported in the context of infection, could the inflammasomes play a role in sensing or in the activation in response to DNA and further, how can I say, release DAMPs in response to tumor cells? It is the first one. And another one, in your point of view, where is pyroptosis in this context of immunogenic cell death?
Pyroptosis. Okay. So inflammasomes. Yeah. The answer. The short version of the answer is yes. They are very important. The long version is there are several ways of activating the inflammasomes, as you know. Data from the Kromer lab when I was still there demonstrated that ATP, I mean, I didn't really go into the detail of ATP signaling happens, but extracellular ATP works in two ways. One is a chemotactic way, so it attracts myeloid cell precursors into the tumor microenvironment, and this is mostly driven by a receptor that's called P2Y2. And when you deplete that receptor, so if you do immunogenic cell death experiments in the absence of a myeloid compartment that express that receptor, then you get lower tumor recognition because the cells don't get into the tumor to pick up the tumor antigens and then migrate to the lymph nodes and stuff. The second way that extracellular ATP works is by triggering inflammasome activation via another receptor is called P2RX7. So when you have extracellular ATP through P2RX7, you get inflammasome activation, secretion of interleukin-1 beta and interleukin-18, both of which are important for the cross-priming of the T cells by the dendritic cells. So you get both a chemotactic and immune stimulatory thing. Now, with the DNA, yeah, there is considerable literature that mitochondrial DNA specifically can activate the inflammasome or can boost inflammasome activation driven by other agents. But we never, for instance, I never studied that from a mechanistic perspective in terms of radiation. I never knocked out an LRP3. I never knocked out caspase-1. I never did one of those mechanistic experiments. We did those in the context of chemotherapy-driven cell death. And we found, again, interleukin-1 blockade decreases detection, and LRP3 knockout mice are unable to get vaccinated, and so forth. So that's pyroptosis. Pyroptosis is my new love, actually. I think it's extraordinarily immunogenic. One of the things is I'm not sure how frequent it is across the board. I mean, myeloid cells for sure. Is it something that how it's gonna go in other cells, right? I know there's this gasdermin, caspase-3, gasdermin E pathway that has been proposed for cancer cells, and it's probably more relevant. But the other, many of the cancer cells actually don't express caspase-1, caspase-4, and 5. So it's a little bit more complicated to go to the standard gasdermin D dependent stuff. The good thing is, at some stage, it doesn't really matter where the danger signals come from, and this is something that has been also underestimated. I mean, we were really, I mean, we are scientists, but sometimes we really operate in a box. First, there's just cancer cells. Tumors are clonal, they are all the same, there's no immune system, nothing going on. And then angiogenesis, and then okay, now we discovered that tumors are vascularized, we have to target the vasculature. And now the immune system, now we only focus on the immune system. But for some reason, we have a hard time doing the links. And I think when you treat a tumor, you treat everything there. You treat the cancer cells, you treat the endothelial cells that make vessels, you treat lymphatic cells, you treat the immune system cells that are there, you treat the stroma, the fibroblasts. The relative contribution of each of these components to danger signaling is completely unknown. And I wouldn't be surprised to find one day that actually fibroblasts are playing a major role. Some tumors contain 50% of fibroblasts. 50% of the cells in a tumor are fibroblasts, and there's very little people around the planet studying fibroblasts. There are some in the cancer-associated fibroblasts. I mean, so I think the danger signal, to go back to pyroptosis, at some stage, if the treatment induces a release of dangers through pyroptosis in the myeloid compartment, I'm still going to be happy if it's not cancer cell.
Cool.
Do I answer was it good enough as an answer?
The next question.
Oh, I was muted. Sorry. Yes.
Okay. Thanks.
Next question, Lorenzo, comes from the YouTube. What do you think about oncolytic virotherapy as a strategy of ICD induction and cancer treatment?
Yeah, I consider it as good as to just publish a review article in Trends in Cancer about that. I think that oncolytic virotherapy and oncolysis in general, the review article was more on oncolytic peptides, but oncolysis in general is very powerful. Because we are I think it's a first, it's a new concept, but it's a first approach to target the heterogeneity of the tumor. Tumors are very heterogeneous, and now we are trying to find out less heterogeneous targets so that we can take into consideration most of the cells. Maybe not all. There's always probably going to be cells that can able to escape. Tumors are very dynamic. Heterogeneity is not only spatial heterogeneity, is also a temporal heterogeneity. So pre and post treatment, the tumors will be different. Even standard evolution of the tumor will generate this kind of heterogeneity. Oncolytic viruses are very powerful for that, and they're very powerful because you can bring in help for the immune system. First of all, they are supposed to mount an immune response because they are viruses, and that would. But you can also engineer them to add features. So the immune system there is both the friend and the foe, because if the immune response against the virus is too strong and too rapid, then you will kill the virus before the virus kills the tumor. But if you're able to allow some degree of viral infection from the very beginning and then boost locally the immune response against the tumor might be able to maybe even eradicate the viral infection later on, but you don't care anymore because the immune response that you generated against the cancer is already strong enough to take over.
And another question from also YouTube is: can cells expose chaperones on its surface in adaptive UPR, not immunogenic cell death, and maybe help it, the response?
Yes, they do expose. Of course, immunogenic cell death is the final result of an adaptation that fails. And many of these danger signaling are actually started when the cells are still adapting. I think from a very humanized perspective is I don't know whether I'm drowning, but I still would want to have the lifesaver closed just in case. So they start emitting this signal to alert the immune system of the potential of cell death to happen. So they start to, expose carreticulin, they start to, which is a chaperone from the ER, secrete ATP. And then just in case, then they fail. But probably there is a communication that operates even when the adaptation is successful. So those cells are not drowning, but they're still in touch with the immune system, again, in case of. And by the way, carotenoid exposure is one of the first events. When we started really the mechanism back in 2009, we determined that in many cancer cell types, carotenoid exposure occurs before phosphatidylserine exposure, which is actually very tolerogenic. So it's a very early event in the process that then leads to cell death.
Lorenzo, I have so many questions. I'm trying to put them in a kind of a logical way here for you. Maybe I'll pick up from something that you already mentioned. So you talked clearly about the regulated form of necrotic cell death. So what about when an accidental necrotic cell death occurs in an individual? Does this reflect an immunogenic cell death? Because, I mean, obviously there will be release of DAMPs. So again, together with these questions comes, the question perhaps, are DAMPs or some classes of DAMPs enough and sufficient, or you need something else? And then now I come back to Karina's question, because also you finally mentioned the cross-priming with the IL-1, IL-18. So do you need something else that is reminiscent of an infection? Again, you talk about mitochondrial DNA. So that's reminiscent of infection. A former PhD student in my lab, Daniel Carvalho, came with a mechanistic answer on the epigenetic therapy of cancer, and that's the awakening of endogenous retrovirus sequences. So how, could you just comment on this more infectious type of signals, even when you irradiate cells?
Yeah. Okay, I will try to address all these questions one at a time. Okay.
I shoot a lot of things.
But
I'm sorry.
I was writing it down.
Cool.
Okay. Accidental cell death. The short answer is actually no. And we tried that by, accidental cell death is something that in the lab we used to do freeze thawing. So you take cells, you freeze your toe, they're breaking apart. So basically, from a component kind of perspective, those DAMPs are there, right? I mean, the caroteculin is not being exposed. Okay, maybe let's assume caroteculin is a bit more complicated, but HMGB1 is there, ATP is there, anything that is released is supposed to be there. And if you do a vaccination with that, it doesn't work. So it tells you it's not just a matter of the component, it's a matter of a spatio-temporal design scenario where the signals are delivered in the right sequence at the right time in the context of a dying cell and not completely dead cells. Even when we did, when you do mitoxantrone treatment, so you treat the cells with mitoxantrone in a plate, in a dish, you use them as a vaccine. If the cells are too dead, so if they have too much PI positivity in that mix, it doesn't work anymore. So again, it's not just a matter of the components, it's really an ongoing process that has to happen inside the mouse. So in the presence of a detection system, which is the mouse immune system. So yeah, accidental cell death in our hands never worked. And we even tried a little bit more refined stuff to complement. So we thought maybe ATP gets degraded too fast, is not enough. We were trashing ATP on top of it. We were trashing recombinant caroteculin on top of it. No, never happened. You need a temporal dissected process for immunogenic cell death to be perceived as such. So it goes down to the other question: is there any DAMPs that are enough? The answer is no. So you need this really like timely process. And also, for a long time, we thought that we would be able to identify a set of DAMPs that are always required. So I don't know, ATP always required for the immunogenicity of cell death. And it turns out it doesn't, we didn't really find that. I mean, in some cases, something is dispensable. ATP, that was my last hope, I prove it to be dispensable myself. All these autophagy deficient cells secrete very little ATP when they die, and still they can be very immunogenic when you irradiate them. Talking to the component scenario, I told you the components, this free thawing, all the DAMPs are there. They are, but maybe part of that timely process is also they get modified. So what you secrete, it's not what you have in the cells because the stress response changed it. I'm thinking about mitochondrial DNA is one of the major examples here. Mitochondrial DNA gets oxidized during the process. And there is a literature from other disciplines like lupus research or inflammatory research that strong activation of cGAS, strong activation of TLR9, which is the other mitochondrial DNA pattern recognition receptor, requires oxidized DNA. So if you freeze thaw your cells and you don't get a lot of oxidation, yeah, the component is there, but maybe it does not show up in the right configuration. And then the infectious thing. I mean, I think it really all goes down to mimicking a viral infection here. The receptors on the side of the immune system were evolved to detect viral and bacterial component. There's no doubt about it. All these toll-like receptor, cGAS evolved as something to prevent yourself from viral infection. When there's the DNA in the cytosol that shouldn't be there, what is that? It's a virus. There's no other choices. Pattern recognition receptor, both the intracellular one and the extracellular one are viral mimicry. And we know that pathogen-driven cell death, so when a cell succumbs to a viral infection, is very immunogenic per se, but in that case, though, the components that are pulled off from an antigen perspective are the antigens that the virus or the bacteria provides. So you go around the problem of having a self scenario. If you take a very healthy cell and you kill it with anything we use to kill immunogenically, radiation, blah, blah, it's not going to protect. It's difficult to demonstrate, but it's not going to generate an immune response because simply you don't have the T cells that can recognize anything in there. So it can become inflammatory because some of the DAMP are inflammatory per se. So you can have an inflammation, but it's not going to generate any sort of immunological memory. It's not going to generate any sort of immune activation, like antigen-specific immune activation. And that is something that I think only applies to perfectly healthy cells, because when you go even to a self cell, so a self that theoretically has been is covered by central tolerance. So this is one of the things. But that cell is sick, for instance, because of, like, I don't know, think about any partially autoimmune disorder like obesity, where there's an inflammatory component. The antigens that are presented by that cells, and so the components of the cells will be modified again by post-translational modifications. So you will have new antigens that are not covered by thymic tolerance that maybe in the context of a cell death will drive an antigen-specific immune response. So even when the cells are really genetically your own, maybe you can end up in a situation where the antigens that they present are not covered by tolerance, and so you have immunogenic cell death in that case. Did I miss anything?
No, I think you were very comprehensive. I just want to go a bit all the way around. So did you ever try to see the effect, if there's any effect on your, let's say, radiation model by knocking down interferon type 1 or IL-1, IL-18 in your setup, and if this has some impact on the immunogenicity?
We never did the interleukin system, so we stayed away from the inflammasomes. But we did interfere in several ways. So we used we did it, well, 3 different ways, I think. We used a double knockout, ATG5 and IRF3. Now, IRF3 is one of the major transcription factors that is involved in cGAS-dependent type 1 interferon secretion. So these cells are actually deficient in type 1 interferon secretion by radiation. And yes, of course, you abolish it. So if they cannot secrete type 1 interferon, you abolish it. And also we did it on the mouse side. So we used IFNAR1 knockout mice. So those lack one of the 2 subunits of the type 1 interferon receptors. And again, you abolish the effect. And we used monoclonal antibodies, again, antagonizing the same subunit. Again, even with that present, you lose all the effects. So the dependency on type 1 interferon is really solid. And this is not only us. I mean, we did it because we really wanted to go like comprehensive, to have the possibility to publish that in a good journal. And eventually we succeeded. This has been published. I failed to mention Nature Immunology last year. But other people demonstrated before us that if you don't secrete type 1 interferon upon radiation, you lose all these subscopal responses.
Cool. Thank you very much.
Sure. Lorenzo, before the next question, I would just add that the person that asked you about the expression in the surface of calreticulin or other chaperones said that she was also thinking about GRP78.
Yes, we never checked it ourselves, and I don't recall. I mean, I know for sure HSP70, HSP90 are exposed. That is being proved by ourselves and others. GRP78, I don't recall seeing that. So it's possible that it has not been investigated, even though it's obviously a very key component of the system, because it regulates the activation of the ER stress response by itself from the very beginning, right? It's a chaperone that takes care of unfolded polypeptides, and when you overload it, then you are basically, that's the way the ER stress response started. But we never did it. We used inhibitors of the 3 branches, and it seems that not all the 3 branches of the ER stress response are equally involved, though. So there could be also a differential effect over there.
Okay. So my question is about the ATG5 knockouts, and from the previous talk by Larissa Cunha, we got to know that ATG5, it's a common part of autophagy and also LAP, LC3 associated phagocytosis. And there are Rubicon that is exclusive for LAP, and OC1 that's exclusive for autophagy. Have you tried to dissect if pure autophagy is the one that is responsible, or there is another way, another responsible that is the LAP system as well?
No, we did it actually. And I mean, I know this is, actually, if you want, also we are working on that, but mostly from the mitophagy side, because we never demonstrated which receptor is involved in the mitophagic removal of the mitochondria. So we do observe mitophagy, but we never dissected it mechanistically, right? So no, we never did it, even though I am aware of the work from Doug and collaborators about the LAP and Rubicon.
Another question. So how important is the cells that are infiltrating the tumor for this Scopal response? Do you have a tumor that's less infiltrated, less responsive to this effect as well? How does it play there?
Well, it's obviously key, and it's key not in the way that, I mean, it's key in 2 ways. So you have 2 scenarios. One is the tumors have a better infiltrate by themselves because they are naturally attracting more cells, right? And that's probably increases the probability of an immune response, a Scopal response to happen. But the model that we use, which is this TSA cells, is a relatively poorly immunogenic tumor. It's not CT26, it's not B16. And in that case, the real difference that how you use radiation to make that happen. And there's a strong work from the De Maria team demonstrating that the number of fractions and the dose per fraction change the capacity of cells, cancer cells, to secrete apointerferon, because you upregulate a nuclease that's called TRX, and that nuclease basically eats up cytosolic DNA when you use high doses per fraction. And it does not get upregulated when you use low doses per fraction, relatively lower. It's not low at all. It's actually high anyway, but it's lower doses per fraction. And that is basically tells you that even if you don't have a strong immune infiltrate up front, because your tumor is not particularly immunogenic, if you play well with the system, so you use the correct dose of radiation, correct fractionation, and a CTLA-4 blockage, then you get an infiltration that is strong enough to mount. Even the initial one was not good. And of course, that's key, because you need dendritic cells to be around. Indeed, if you do the experiments in BATF3 knockouts mice, doesn't work. BATF3 knockout mice are lacking a specific subset of dendritic cells that is got good cross presentation. So you really need dendritic cells to be around. In the context of chemotherapy, we never tested with radiation, but in the context of chemotherapy, lymph nodes seem to be not 100% required, because there is tertiary lymphoid structures inside the tumors that can operate for as sites of cross priming. We did in the past, we did this immunity paper 2013, we were doing lymphadenectomy. So we were removing the tumor draining lymph node, all the chains, and we would still get the vaccination effect, from the local tertiary lymphoid structure. I don't know with radiation. So you need these dendritic cells, key, and then you need, of course, CD8 positive T cells, and you need for chemotherapy, interleukin 17 producing gamma delta T cells. In many disease settings, actually, we also have data in that they seem to be immunosuppressive. But when you do the vaccination test in mice where the interleukin 17 system is blocked, then you don't get vaccination. So there's also that component. And then, of course, basically that's it. So you have the full circle. You need an antigen-presenting cell that is good enough. So BATF3-dependent type 1 dendritic cells, or CDC1s.
Right.
You need CD8 positive T cells, and you need interleukin-17 producing gamma delta T cells.
So using the same line of thought, you would say that a tumor that has a bigger mutation burden would be better for this type of vaccination?
Well, I think the answer is yes, but I think also the tumors that we use, even those with the lowest mutational burden, especially in the vaccination setting, which is a very sensitive test, as I mentioned, there's enough mutation anyway. So the spectrum of mutation is very different in mouse tumors as in human tumors, but even when we take the leastly mutated ones, it's enough for the vaccination test. Now, from the human perspective, so we move away from the setting of vaccinating people, because this is not what you do, you treat people. The mutational burden has been associated with increased responsiveness to several immunotherapies, as much as, for instance, you now measure mutational burden to potentially use those therapies. The point is, sometimes mutational burden is associated with a tumor heterogeneity that will actually at the end be against treatment. So I think because the more heterogeneous a tumor is, the more possibility that one of those cells will be resisting any treatment, right? So there is for the mutational burden / inter-tumor heterogeneity setting, I think there is an optimal level. If it's too much, then the evolution is too favored. So you can, if you have a billion clones, one will survive and will drive result. If it's not enough, then the immune system will have a hard time finding something to work on, I think.
Excellent, Lorenzo. One very quick, technical tip. When you prepare your dead cell samples, So imagine, then you said you want to get rid of the chemotherapeutic drugs, whatever. I mean, you just centrifuge, because I know that cells lose the buoyancy a bit. So you just push them to the bottom like strongly, or is there any other like?
No, that's it. You wash, but you wash. I mean, we used to initially, the initial test with immunogenic cell death was actually cool because we were using doxorubicin, which is red, and mitoxantrone, which is blue. So basically, we just washed them until the pellet was not colored anymore. That was but for other drugs you cannot, of course. But what you can do is, I mean, in our hands, 3 washes in PBS are good enough. Were good enough to wash out.
And because then you work with whatever is precipitate.
Yeah, you spin faster. You don't spin a standard because you will lose the apoptotic bodies. You spin a little bit faster, not to smash everything down, but 1.5 times the normal speed, something similar, so they will not die. Whatever is not dead will not die, and you will recover all the apoptotic bodies.
Cool. And then you just adjust by the number of the cells that you start. You don't really matter if you end up with more or less protein or stuff like that. Doesn't matter.
We inject a number of cells, so you count the cells, and we normally check for PI positivity, for what I mentioned before, because if there's not enough PI positivity, those cells we do a tumor. And it happens. So sometimes you just get a tumor at the vaccination site, and you know that your killing mode was a little bit lousy. And on the other hand, if there's too much death, probably you're already in this scenario where it's too late. You will not have this timely process in the host to generate a vaccination effect. So we checked, and we normally set ourselves in between 50 and 70% PI positive cells. So that is a place that we like. I'm not saying that it will not work with lower or higher, but I'm saying if it's too low or too high, it's not going to work.
All right, that's very important, because some of the setups we tried and it never worked. So I know that it has to be specific.
Yeah, and also on the challenge injection. So when you challenge later on to test, you normally use the so-called minimal dose. So the dose that in non-vaccinated animals is the lowest to generate a tumor, because of course if you put 10 million cells, they will generate. I mean, it's not. You know what I mean, right? So you have to.
Yeah, no, but it's important to mention to the students and everyone.
Of course.
All right.
So can I just make a quick question? It's more like a philosophical, fun question. It's about you start the lecture saying that the definition of cell death is rupture of the cell membrane, right? The plasma membrane.
Operationally, yeah.
Yeah, operationally, right? And then apoptosis, physiologically, you wouldn't see that in vivo, right? The cells would be removed much earlier than this opening of the plasma membrane. So would you consider more apoptosis as a program to facilitate cell removal, efferocytosis, and communication rather than a cell death program?
Yeah, absolutely. For actually, fun because I had this conversation in the bus this morning with a friend of mine. I mean, I know people think, and they have material to think that, for instance, caspase 3, 7, and 6 are executor of cell death. So that when you remove them, the cells don't die. There is a literature out there, and we have our own data in here that it's not true. What happens is they die at a different kinetic, and they die with a different manifestations, including immunological correlates. And we published it in a very low-profile paper last year, but I wanted to have it out of my desk. It's an oncoimmunology paper where we basically compared the immunogenicity of caspase 3 knockout cells to wild type, again, these breast carcinoma cells. And of course, if you look at the percentage of cells that are PI positive 48 hours after radiation, yeah, the caspase 3 knockout is lower. I mean, no news. It's like everybody did it before us, right? But when you go for clonogenic survival, So when you go and measure 14 days later, how many cells were able to form a colony, it's exactly the same. So this talks to two things. One is the connectivity of the system, and the other one is many of the players that we believe to be critical for executing, I think they're not that executors. They're just regulators of the interaction with the immune system, namely efferocytosis, induction of immunogenic, well, perception of immunogenic cell death, you name it. And if you think about another fun part of it, the classical definition of caspases is there are cell death caspases and inflammatory caspases. I'm not really sure about that. I'm sure that there are inflammation regulating caspases. They are all inflammation regulating caspases. Some does it in one way, some does it in another way. Caspase 11 drives inflammation because it triggers pyroptosis. Caspase 1 cleaves interleukin 1 beta. Caspase 3 takes it down, makes the cells die later, cuts gas. There's a lot of so it's just two facets of the same process to me. It's not two different processes.
It's great because this is what Seamus, speculated in the first lecture. He also goes in the same line of you, saying that there's no apoptotic caspase. It's just pro-inflammatory and anti-inflammatory caspase.
Totally agree.
Something like that. And we also published a review, in a even lower, impact journal, saying that apoptosis is a cell death program or a cell disposal, only to dispose the
yes,
corpse that are ready. So that's very good. All right, man.
I think Karina has a question, Gustavo.
Oh, sorry. No? Anybody, you guys,
Sorry.
Students here at the Zoom?
Yeah, actually, I do have a more hypothetical question.
Okay.
I was wondering, because you have shown immunogenic cell death more like, at least in your lecture, more like something that we could use, an approach that we could use to treat like tumor. But I was wondering if we could also use it as an approach to generate more longer immune responses, such as thinking like preventing infections, and perhaps, I don't know, something more related to memory immune response generation or something like this. So if you have any comments.
Well, if I understood what you mean, it's difficult to put it in place in the sense that we have very strong and potent tools to generate immunity to pathogens. At the end of the day, immunogenic cell death is a way for the body to generate a vaccine, like a fact. And I can tell you the adjuvants that we use in vaccines, they are potent enough, or actually probably even more potent than whatever comes out of a cells when it dies. The possibility that, immunogenic cell death can be modulated two ways. One way is I want to have immunogenic cell death because I want to cure cancer, which is what I'm trying to do here, not curing cancer, but having immunogenic cell death. But the other way could be, I want to inhibit immunogenic cell death because it contributes to autoimmune disorders, or it contributes to many pathologies that are actually, this is a relatively new perception, but there's dozens of pathologies that have not linked to an immune response that now they start to be. Myocardial infarction is, of course, you get a flow, interruption in the flow in an artery, and then part of your myocardium die. And there's a lot of people who investigated the mechanisms of that in that. And that's fine. But there's now data from preclinical models and patients that if you block the immune response that cell death generates, the patients will do better. Of course, there's always this spectrum of severity. A very severe case will die anyway. A very mild case will recover anyway. But there is a window of opportunity where modulating, for instance, type I interferon might provide an advantage. Now, is it possible in patients? I still don't know, but it's for sure something is going to be investigated. And also, and this kind of non-expected links will continue to come. So there's going to be other pathologies where you're going to see some degree of perhaps not even immunogenicity, but some degree of autoreactivity. So your cells are mildly more susceptible to generate an immune response, to generate inflammation, and that is contributing to the pathology of the disease. In a sort of obesity-like scenario, if you think about obesity, there is a inflammation in the adipose tissue, which contributes to the disease. And do we know whether immunogenic cell death is going on in that system? We don't. So maybe it's an interesting field to develop.
Yeah, I must say that I really like Anna's questions. I may approach in a different way. Some pathogens evade immune response by switching the differentiation of effector cells toward the suppressors or regulatory cells. So have you tried to look at any way on, the differentiation of TH cells, in the presence of immunogenic cell, that different forms of immunogenic cell, that or something like that? Does it counteract, the cytokines that would push towards regulatory cells or something like that?
We never did it in the sense of really dissecting the contribution, especially if you think about the vaccination effect is really a potent Th1 response, right? That's really a vaccine kind of scenario. If you think about inducing immunogenic cell death in situ, so when the tumor is already in place, so the tumor in place, some of them at least have a high Treg population, they have a high macrophage population that is immunosuppressive, myeloid-derived suppressor cells. So I think when we induce immunogenic cell death in that scenario, it's a very complex. First of all, it's way more complicated. So to get this abscopal response is really complicated. But it's a mixture of effects. It's a mixture of effect that goes from, okay, first of all, you jumpstart a tumor targeting immune response. You need that. But to have that, perhaps you need Tregs to die. And for instance, when you do it with radiation, we know that Tregs are relatively susceptible to die with radiation. So we are, and the other cell types might behave the same or completely different. But the concept I want to pass here is that in situ, is not just my cells are dying in an immunogenic manner. And I also have the T cells around that work. Yeah, maybe that's not as complicated as we think. In situ, though, there's going to be a line of other problems that we will have to take care of to make it really immunogenic. Again, Treg depletion, TAM2 depletion. Sometimes some tumors are really, they are really difficult to infiltrate. Like they have this strong stroma. So if the T cells that I generate with my immunogenic cell death at one site will not be able to infiltrate another tumor because it's too thick, the stroma, then it's still not. So the barriers are in the real world are really many more than just there's antigens and there's T cells and there's adjuvants.
All right, everyone. I think we run away from our time. We already passed. I'd like to close up by thanking Lorenzo so much for this brilliant talk. It was great to have you here.
Thank you.
We hope that you can also accept our invitation and talk at our program at the weekly seminar of the Department of Immunology. More
with pleasure.
And yeah, let's keep in touch.
All right.
Okay.
Thank you, everybody.
Thank you so much. Guys, don't forget the students and the one interested in tomorrow's 2 lecture. That will be in the afternoon from 2 to 4 and 4 to 6 p.m. Brasilia time. And Lorenzo also, if you want to join us, if you have time, you're more than welcome to participate at our discussions.
Thank you. Unfortunately, we'll not be able to make it tomorrow, but we'll be in touch.
All right, man. Thank you so